My name is Rodney Miles. I'm an associate professor of pathology at the University of Utah, and my clinical practice is focused in hematopathology, which is the diagnosis of leukemia and lymphoma, and I'm particularly interested in pediatric leukemias and lymphomas. From a research perspective, one of the things that I'm most interested in is why some lymphomas relapse and don't respond to therapy, while other lymphomas of the same type will. Pediatric non-Hodgkin lymphoma is an important cancer in children, adolescents, and young adults. It's a curable cancer, but there are still some patients that relapse or whose disease does not respond to chemotherapy, and those patients do very poorly. And we don't understand the difference. As a pathologist, when I look through the microscope to make a diagnosis of lymphoma, I can tell what type of lymphoma it is, but I can't tell if it's going to respond to therapy. So that's something that we've been studying in my research lab here at University of Utah, working with lymphoma cell lines, where we're manipulating genes to see the effects on how those cells will then respond to chemotherapy. And we found that by altering some genes, we can make lymphoma cells more sensitive to chemotherapy. So this is very interesting, but it's most important to be able to take these types of findings from cell lines back into patient samples. So I work with a number of collaborating individuals who are also physician scientists interested in pediatric lymphomas, and they're from multiple institutions across the country. And we're all focused on this translational research in pediatric lymphomas, where we're trying to learn things in the laboratory that then can be translated into the clinic, leading to better patient outcomes. One of the limitations that we all face is that we want to take the findings from cell lines in mouse models and see if they're relevant in actual patient lymphomas. So lymphoma, despite being an important cancer in young patients, is uncommon because all pediatric cancers are uncommon compared to things like breast cancer or prostate cancer or colon cancer in adults. So no one institution can gather enough pediatric lymphomas of any subtype, especially to include relapse samples. So what we need to do is work together to pool our resources in a way. So what I've proposed to do, and what the Pediatric Cancer Research Foundation has generously agreed to support, is to open and maintain a pediatric and adolescent and young adult non-Hodgkin lymphoma biorepository. So what we will do is collect lymphoma specimens from patients from our institution as well as collaborating institutions from across the country. And this won't require extra biopsies. We'll use tissue that's left over after the pathologist has made the diagnosis. These tissues will all be collected centrally here in my laboratory, and they can be made available then to pediatric lymphoma researchers to validate their cell line and mouse model findings in actual patient samples. So this biorepository requires skilled technologists who can be here to receive the samples and ensure proper handling so that they're not compromised. We'll isolate DNA and RNA from these samples, and we'll isolate viable cells and cryopreserve them so that they'll be available for future studies. This will also require, of course, careful cataloging of de-identified clinical data, like the type of lymphoma, whether it's a primary diagnosis or a relapse sample. So the goal going forward is to have a repository of these biological specimens that lymphoma researchers like myself and my collaborating colleagues can use to validate their findings, improve that what they're seeing in disease models is actually relevant in patient samples as well. And it's through this type of translational research that we're all striving to improve patient outcomes, ultimately by identifying prognostic features, therapeutic targets, and other ways that we can improve therapy for young patients with lymphoma. And that's really the goal of what we're trying to support here.